Hello, everyone. Welcome to the first current topic session of third term. For our course enrollees, please remember to sign in via the Course Plus tracker. That's for folks in the room and folks joining us via Zoom. It's a tab in the Course Plus site, and you just mark yourself as attending. And I'm going to pass over to Dr. Priya Dugal to get us kicked off.
Hi, good afternoon. Not sure where I'm looking, but we are excited today to have a very illustrious panel of our own faculty who are going to highlight some of the studies that have been led and housed here at Hopkins for, a quarter of a century, if not longer. I think actually 30 plus years, 35 years for some of our studies, and really give a breadth of several different types of work that you can do. We're hoping that this is an opportunity both for students and for faculty to think about things that they could work on in these studies, that they could collaborate or co-investigate if they're a faculty member, but really ideal for people if they want to pick up a study as a doctoral student, either as part of their dissertation or something else, or for a master's student, or just another project. New ideas are born from these types of things, and so we hope that that is what will happen. So our first speaker today is Dr. Christine Latacasta, who's going to talk about neurodevelopmental studies that she leads.
Great, thank you, Priya. So I'm actually in the genetic epi track, for those of you that don't know, but as Priya said, I'm going to be focusing and highlighting some of our autism epidemiology studies in the department that were established here and are co-led by myself and others across departments. Where's our advance? If I can figure this out. Not advancing, Laura. You know what? I think I can just do it with the, see if I can do it this way. Like nothing's working.
The clicker was working earlier.
There we go.
Just
Just slow. Okay, great. And so what I love about these, actually, what you'll see is I think they really span, you'll see from the past to the present, and then also from cells to society. So really ranging from biologic mechanisms all the way through practice kinds of opportunities. And so much of this work is also housed, and I wanted you to be aware of the Wendy Clagg Center for Autism and Developmental Disabilities, which is the center in our school as well. And so the first one that I'm going to highlight is called ADAM for short. It's funded by the CDC. And ADAM was established in our department over 20 years ago, actually, by Dr. Craig Newshafer. And then from 2006 to 2021, it was maintained by Dr. Li-Ching Li. And then I'm thrilled that Dr. Elise Pass in mental health and I were successful in our renewal last year. And that work will continue for the next 4 years. And what this study does is it's the autism surveillance study. So if you've ever heard of those numbers of 1 in 44 kids or 1 in 50 kids has an autism in Maryland, this is the study that produced that data. It's one of the original sites. As I said, it started in 2000 and is one of the only sites that has had continuous prevalence monitoring of autism for the past 20 years. It computes the prevalence of and describes the characteristics of 4, 8, and just this year, the new piece of that will be in 16-year-old children as well in Maryland. And it also allows us, because of this long history, to look at trends in Maryland over time. And then the results for this are really very practice and translationally focused. And so they inform stakeholder resource planning, right? So how many clinicians, how many resources do we need in the state of Maryland to address early identification and other service needs of people with autism? So what kinds of opportunities then are there for you all in this? So as you may realize, it is this big public health practice and stakeholder engagement component. So we develop lots of communication materials and approaches to really effectively inform ASD public health policies in Maryland specifically. So we have a wide range of different policymakers, nonprofit organizations, and then snapshots and materials that we distribute to the communities. In addition, there are opportunities for Maryland-specific data analyses from the data that we're abstracting to answer public health questions. So I wanted to give you a flavor of what that might look like. One of our students used that data to ask about the influence of sociodemographic factors on the timing of ASD diagnoses. Okay, so that was study one. The second study, with a long-standing, again, history and established in our department, is called SEED for short. The SEED study is a large multi-site autism case control study. And so we now have almost 7,000 families that enrolled across 6 sites in the US. And the Maryland site specifically brings really strong genomic expertise to that network and is also the home of the biorepository for that network. And so it again was established by Craig Newshafer and Danny Fallon in this department and maintained for the past several years. And then currently we have sort of new leadership, myself, along with Dr. Becky Land at KKI and Heather Volk, and Home Varsity again here in our department overseeing the biorepository for that study. To give you a sense of what the study design was, is there were 3 phases of original recruitment in SEED and enrollment. Children that were between the ages of 2 and 5 were enrolled, and then they ended up in one of 3 sort of buckets, and it's about a third distributed. They're either defined as an autism case, population control, or they have a developmental disability other than autism. And currently, we received new funding to continue the work with SEED, but now we're doing follow-up studies. So we're not newly enrolling kids age 2 to 5, but we're now following up those original participants now that they're in their adolescence and early adulthood to collect new information and new data about them during that life stage. Okay, so what kinds of data are available in SEED? There are a whole host of different socio-demographic and baseline characteristic data that were collected. We have a number of different outcomes that were collected at age 2 to 5 in these children, including really rigorous developmental clinical phenotyping. I've listed a number of them here. There's also now pilot data and new data being collected in adolescents on outcomes. So that'll be an area moving forward that we're eager to examine as well in terms of outcomes, and then changes, right? And some of these outcomes across time we can look at as well. There's robust prenatal environmental exposure data that was collected in all of these participants across many different kinds of exposure domains. So everything from behavioral kinds of exposures to medical conditions, pharmacologic use, infectious disease, among many others. And then finally, we have omics data. So for almost all of the SEED children and many of the mothers, we have genome-wide genotyping data, we have whole exome sequencing data in a subset of trios and ASD cases, and then we have DNA methylation data among a subset of participants in SEED1. So what kinds of opportunities then might there be? I wanted to give you a flavor of what our research priorities are and some of the questions you might ask. The first is really to use some of the extant data. And again, our Maryland site really focuses on the genomic perspective here. And so we want to leverage that data to address our priorities. And our priorities are to identify biologic mechanisms that could serve as targets potentially for interventions. We want to help inform what I'm calling precision public health efforts, right? Are there subgroups or subpopulations that we can identify that might be particularly susceptible to modifiable exposures or certain outcomes based on their genetic composition or context? We want to build and evaluate biomarkers. So can we use omics data to accurately predict their short or long-term health outcomes? And then finally, there's lots of, as you can imagine, method development work as well to overcome many of the challenges we face when trying to address and answer these scientific questions. Okay, so finally, the third study. This is a new one I just wanted to highlight because we're really excited about it. This is a new project that Heather Volk and I received last year, which is an Autism Center of Excellence. And the goal of this project is to establish a network to evaluate gene-environment interactions in ASD. There is an observational epidemiology component. We've somehow convinced 18 sites to work with us from around the world, where we have over 175,000 total participants and over 40,000 autism cases. We have a data coordinating center and many other kinds of committees and teams. There is a second branch of this project, which involves experimental brain organoid model systems to complement some of our human work. That work is being led by Thomas Hartung and Lena Smirnova in the Department of Environmental Health and Engineering here. And really, I think, important and exciting for this project is that we have an entire community advisory board and outreach aim and goal where we really want to have a two-way street with this network with our stakeholders. We want to understand, right, what their needs are and make sure that our research goals align with their needs, our autism stakeholders. And then we want to best take what we're finding and learning and be able to translate to them in a way that's meaningful and important to them. So we're really excited. There are lots of going to be opportunities moving forward, again, for faculty as well as students to be involved in this network. You can lead method development projects and initiatives. I've left listed just a few here: statistical method development, missing data issues, and then really accommodating diverse and admixed ancestries is going to be a big challenge in gears. And then you can also lead projects to answer specific scientific questions as well. And I've just given you a flavor here of what some of those questions may be. So I talked a lot about autism. I focused on those, but being in the genetic epi track, there are lots of other, I think, omics opportunities out there in the department for many of the studies you're going to hear about today. So this is just to say, stay tuned, because I know these studies at least have some omics data, and I'm sure many others do as well. So stay tuned for their talks.
All right, so thanks, Chris. So I'm Shruti Mehta, and I'm a faculty member in the Department of Epidemiology, and I'm going to talk today about the ALIVE study. So I've had the good fortune of co-leading the ALIVE study with Greg Kirk since about 2004, but the study existed for a good 15 years before we came to it. So it was started in the late 80s by the visionary Frank Polk with his then doctoral student, David Flahoff. And we have, like all of these studies, a number of collaborators, both within the university, in and outside the department, across other schools in the US and actually across other countries. What I've shown here are just some photos of the current collaborators within the Department of Epidemiology, so you can get a sense of what other faculty are working on this study. So ALIVE is a study focused on people who inject drugs. The original objectives in the late 80s were to characterize the incidence and natural history of HIV infection among people who inject drugs. So something we know a lot about today, but back then really knew nothing. And it was truly the only study of its kind at the time. And I think today is the longest running cohort among people who inject drugs. So we are a fixed site, single site study. So we're in Baltimore, Maryland, and we see participants out of a single study location. We're community-based entirely. So our recruitment is street-based community outreach and word of mouth, and our follow-up is semi-annual. So this gives you a little bit of a picture of our recruitment over the years. So the first grant was written in 1986 to recruit 640 people inject drugs who are living with HIV and follow them for 5 years. And then the doors opened in 1988. We have moved clinics, I think 4 or 5 times over the years. We've opened enrollment 4 times. We shut our doors for the first time, like many other studies, in March of 2020, but we opened them up as quickly as we could in December of 2020. And we're about to start recruiting again in a couple of months. We have recruited across all of these periods about 5,500 participants and followed them for more than 35 years. So this just gives you a little flavor of the population that we've seen. So when ALIVE started, so this shows all the different recruitment cohorts at the time of enrollment. When the cohort initially started, it was predominantly male, predominantly Black, 35 years of age on average, and about 30% HIV positive, and everyone lived in Baltimore City, sort of by design. And what you see here is the evolution over time. And in part, this is the evolution in the opioid epidemic in Baltimore. So we see an older population, we see increasing numbers of women, we see fewer Black participants, and evening across other races. And we also start to see people enrolling from outside of Baltimore City. Like all the studies you're going to hear about today, we collect a lot of data. Our participants, our struggle is always, how do we keep the visit, under 2 to 3 hours and not drive everyone crazy, but collect as much as we want to. And so here I've just given you a flavor for what we collect at a single semi-annual visit. So we have behavioral surveys that are administered by interviews where we collect not just information about substance use and risk behavior, but also things like depression, quality of life, healthcare discrimination. We collect a detailed self-reported health history, so not just related to HIV and other infectious diseases, but comorbid conditions, as well as engagement and care for these conditions. We geocode everyone's home address so that we have spatial and neighborhood level data. We collect a specimen, a laboratory specimen at each visit. And what you see here is the testing that we do at a single visit. So HIV-related testing, also testing related to chronic comorbidities, but then we maintain a large repository of sera, plasma, and cells. And we have countless other tests available from various visits, everything from inflammatory markers to genomic data to epigenetics. Our clinical assessments have evolved over time. What we're doing currently is essentially vital statistics, general body mass index measurements. We do a FibroScan assessment to assess liver stiffness. Hepatitis C is a huge comorbidity in this population. We do spirometry for pulmonary function, and then we do some aging assessments, so functional status assessments and a frailty assessment. And then finally, to complement the self-reported, you can imagine the challenges with self-reported medical history. We also do a number of record linkages to document outcomes. So everything from cancer diagnoses to other clinical diagnoses and linkages with the National Death Index. So big strength of ALIVE and like all of these studies is just this incredible wealth of longitudinal data over these years. And what you see here is just mortality trends across the 35 years of the cohort. But just to give you a flavor, I think this data is as of July of last year. So we've had just under 74,000 person visits, just over 850,000 specimens in the repository, a median of 7 visits per person, 369 HIV seroconversions, and the most sobering statistic is that nearly half of our population has died across follow-up. So our aims are really see ALIVE as a platform for other work. So ALIVE creates the infrastructure upon which other investigation is built. So these are the core aims of ALIVE today. So really we have 2 areas of focus. The first is related to opioid use. So temporal trends in the epidemic, the impact of those changing trends on outcomes like overdose, HIV seroconversion, hepatitis C acquisition, and mortality. And then the other aspect is related to aging, right? Our cohort is getting older, people with HIV are aging. So looking at the links between HIV and engagement in care for HIV and chronic comorbidities, and that their collective impact on morbidity and mortality. But like I said, this really just creates infrastructure for other investigators, students, and fellows to build upon. And so what you see here is just a collection of some of the currently funded sub-studies that are built, and they leverage the infrastructure, both in terms of the data collection that's going on, the specimen repository. It can be a base for people to analyze existing data, add a question or questionnaire to the existing instruments, or do actual clinical assessments, right? And you have the base infrastructure taken care of, so the costs are a little bit less to add on. So it just gives you a sense. So the ones in orange are disease-focused investigations. So we have respiratory, liver disease, frailty, and a new oral health study. Greg's been wanting to do this for years. Oral health is one of the biggest challenges for our participants. We have basic science investigations, so you can see the immunologic grant. We have a pharmacogenomic study, some very behavioral assessments that dig a little bit deeper into substance use, polysubstance use, and a couple of grants focused on COVID impacts being led by Becky Jenberg. And then the last 2 in blue are training awards. So we have an F31 active right now for a doctoral student here, and then a training grant that Greg leads. So specific opportunities, there are many. I just highlighted some of the general areas where we're looking for people to come in and take advantage of the data and what's been proposed. But really it's open, right? As I said, you can ask a question with the existing data, you can integrate new data collection, and you can do that through, with or without funding. You can come in and seek to do something with what we have or write a proposal. The other thing that I'd say, particularly for the students who are hopefully online, ALIVE is here. Right? Our site is down the street. So you can go to the clinic anytime. And I think that's a huge advantage, right? So it's one thing to get the data, but I'm a huge proponent. If you're going to work with this population, if you're going to work with this data, go talk to some participants. And so we really highly encourage that, insist upon it for our students, but it's a tremendous opportunity to have the site right here. So for those of you who are interested, the best way to get involved is to email either Greg or myself. And just want to acknowledge, I already acknowledged the investigators, but the study participants and study staff who have generously donated their time for years. Some of our study staff has been here from the very beginning, from the days the doors opened. So thank you. Is it on?
think I got it.
You got?
Is that good? Nope, it didn't show. Hold on, we'll get it.
So if you
Uh-huh.
move them, you want me to?
Yep.
If you it's a, I can't, I got, there we go. Yeah. I never
There we
thought-
go.
wanted to be IT,
And
but
to advance.
all you have to
This
use the
one.
clicker.
Okay.
I don't know what.
Hey, everybody, I'm Amber D'Souza. I'm in the cancer epidemiology group, but also the infectious disease epidemiology group. And today I'm going to speak with you about one of our cohorts, the MaxWise combined cohort study. I actually did my PhD here and learned about these cohorts while I was a student and got very excited about becoming part of a big collaborative cohort study, getting involved, and became involved as a junior investigator and then senior investigator. So hearing kind of what Trudy said and what just everybody said, it really is a fantastic opportunity to get involved in these cohorts, and they're career makers. So just to give an overview of our cohort study, we have 13 study sites across the US. This is a combination of two cohorts that were separate before. We had a cohort of gay men and we had a cohort of minority high-risk women, and they were combined a few years ago. So we now have 13 study sites. We have enrollment every few years, and together we're called the MaxWise Combined Cohort Study. So the study of men started in 1984, before there was even a test for HIV. Gay men from across the US were enrolled to try to understand what was affecting them and causing AIDS. The women's study was started just 10 years later. We've had, again, enrollment throughout. There's been just continual investments and advancement of the science, but you can see we've now been going on for more than 35 years, semi-annual data collection in these participants. So currently there are more than 4,000 active participants. There have been more than 10,000 participants who have participated across the years, and we have all of that data and the biorepository of all of those samples. There's been more than 3,000 publications. As our cohort has aged, we have an increase on aging with HIV and comorbidities within HIV, but we also are enrolling people who are in their 30s, as part of the Southern HIV epidemic. So we really have participants across the spectrum. This is looking at our current funding for our current 8 years. You can see right now we're in what we're calling V103. We restarted at Visit 100 once we merged. But we have all this historic data for the 35 years of historic data that continues to be an incredible repository of analyses. And we now also have this prospective new data ongoing. What do we have data on? Well, really, it's very similar to the other cohorts that I've talked about. We have our core protocol where we get our biological samples and we get our surveys, and we have, assessments for pulmonary and cardiac and fibrotic function and kind of all sorts of different organ systems. We have medical exams. We have self-reported personal, private stigmatizing behaviors. We have outcomes where we do adjudication. We have passive surveillance with registry matching. So there's quite a lot in our core protocol. And then we have lots of nested grants ongoing. You don't even have to be at one of our study sites. So our grants are international. Anybody can get research approved and added in. Okay. So a little bit of our demographics of who we have. So carryover are participants who were in the separate Max and Wise cohorts and have now re-enrolled in this combined. And then of course we have some new enrollees. So we're up to actually now, since we did this report, about 1,000 new enrollees so far in the past 2 years, another 3,300 carryover, so a little over 4,000. You can see that around 2/3 of those are people living with HIV, and 1/3 are similar people without HIV used for comparison. About half the cohort is, and this should not say cis male, sorry, that is a mistake. These are mostly men who have sex with men. And then about half the cohort is cis female. We do have a small number of trans individuals as well. And the median age is around in the 50s, but we have, and this is the interquartile range, lots of people in their 40s and 60s. We also have 95-year-old enrolled, and 30-year-old enrolled. So we have, the outliers as well, but most of our population is in this 40 to 70-year-old population. Here's the age distribution of our people living with, this is by sex and by HIV, so that you can see we're not good at studying HIV in young people. The numbers are very small there. But for ages 40 and up, which is why we really focus on aging with HIV, we have a lot of individuals. In terms of some of the other areas that we collect a lot of substantive data, there's a lot of cardiovascular and pulmonary disease measures we have. We have sweep quality assessment. We have tons of neurocog data. We have data on frailty and physical disability, cancer data, pathogenesis data, metabolic liver and kidney dysfunction, psychological and behavioral health, and health disparities. So welcome involvement. Like any big cohort, it does take a little while to get the data. So for students planning projects, you do have to plan in time to propose and get the project vetted and get the data. But, you know, we welcome people in all stages to come and get involved.
So hi, I'm Lisa Jacobson, and I'm one of the PIs of the ECHO Data Analysis Center. ECHO is the Environmental Influences on Child Health Outcomes. We're going to go from studies that have been on since the 1980s to now a study that was started called prospective or active data collection as the ECHO study, which actually started in 2016 was funding. I want to introduce Dr. Monica McGrath. Monica is got her master's here and then her doctorate at Harvard. And she is the director of our ECHO Data Analysis Center. And so she's going to present on the ECHO study.
Thank you, Lisa. So we're going to shake it up a little bit, and this study is focused on children. For additional information, we have two websites here. One is our website that we have on the Maine School of Public Health, talking about the methodology work that we do here at the ECHO DAC, and then one, a public-facing website that you can learn more about ECHO. Okay, so what is ECHO? The ECHO's program objectives is to improve the health of children and adolescents by combining observational as well as interventional research that inform high impact programs, policies, and practices. We attempt and hopefully are successful in terms of instituting best practices for conducting team science, that we take advantage of the multidisciplinary nature of the work that we do. And the ECHO mission is very lofty, is to improve the health of children for generations to come. So what does that mean? What is our actual scientific goal? So our goal is to understand the effects of early environmental exposures on child health and development. We define early from preconception to approximately age of 5 years of life. And what do we consider environmental exposures? We consider anything that you believe or can hypothesize that relates to child health and development. We collect it. We collect it using surveys and specimens and addresses and everything that you can imagine. We have a lot of it. So that includes physical and chemical environmental exposures, air pollution, chemicals in our neighborhoods. We have information on societal factors, stress in the home, discrimination that we may experience, social factors, SES-related measures, family dynamics. We have sleep, we have diet, we have physical activity. And I think as Dr. Latacasta alluded to, we also have epigenetics and we have genetics and we have microbiome. The goal here is to develop research questions that will lead to impactful interventions, policies, programs, and practices. For organizational purposes, we group our interests into 5 main outcome working groups. We have pre, peri, and postnatal factors. That's looking at preterm birth, low birth weight, birth defects. We have upper and lower airways, which is respiratory infections and asthma. We have obesity, overweight, how children grow, as well as various metabolic disorders. We have neurodevelopment, which includes autism spectrum disorder, ADHD, cognitive development, executive functioning. But we're also interested in children's positive health and well-being, recognizing that children may have a chronic disease, but what resiliency factors can help increase life satisfaction and meaning and purpose. So what is ECHO? ECHO cohort utilizes a collaborative study design. We have 69 cohorts that participate in the ECHO program. It is a network of existing observational cohorts as well as clinical trials. All of these were existing when they joined the ECHO program, so they captured data with different research protocols. Within ECHO, we established a new protocol which established a set of common core elements and outcomes of interest that unite these existing studies. ECHO was also established to facilitate collaboration with investigators, as well as all types of investigations on childhood health, and not designed to answer one specific question. The sort of way we talked about earlier, sort of a data resource platform. The goal of ECHO is also to do solution-oriented research, and we involve stakeholders as well to help us inform our research, to develop policy, programs, and practices. ECHO offers many, many challenges. I will not lie, but there are also many, many exciting opportunities that exist with using our data. So to describe our cohort a little bit more, as I mentioned, we are 69 pediatric cohorts across the United States as well as Puerto Rico. We have over 90,000 participants, which include biological mothers, biological fathers, adoptive parents, as well as caregivers, including grandparents. We have almost 60,000 children. We have over 25,000 of them in active follow-up, and tens of thousands of biospecimens. And you can see sort of the geographical distribution of our cohort sites. We also are consistently striving to ensure diversity across our program in order to represent as well as to serve all of our families. Approximately 45% of our individuals identify as non-white, and we have about 25% of our population as Hispanic. So Dr. Latacasta alluded to that we also have genetic data. We have genetic and epigenetic data. We have genome-wide genotyping. We have genome scale epigenetic and a whole host of opportunities for both of these. We also were able to pivot successfully in terms of creating research opportunities for COVID. In September 2020, working with NIH, we were funded for 6 COVID-19 projects, and they are listed here. How does COVID-19 amplify racial and social inequities? How does COVID-19 infection and maternal stress affect a variety of childhood outcomes? In addition, we do a lot of other research. So right now, the ECHO Data Analysis Center is working on over 150 different research projects, which span numerous exposures, numerous outcomes, all focusing on pregnant individuals and their children. Here is an example of some of the ones that we are looking at. Number 1, childhood asthma and neurocognition. 2, prenatal pesticide exposure and airways outcomes. Breastfeeding in terms of child obesity. Air pollution in terms of blood pressure. And as you see, the list goes on and on. So we have at the ECHO Data Analysis Center numerous opportunities for both students and faculty. We have student research assistant positions available. We have thesis opportunities. One thing I wanted to mention is that we are not located here in the main building. We are located down in Candler, which is a beautiful building to visit and work at. If people are interested in any opportunities here at ECHO at the Data Analysis Center, our contact information to please contact Kristen Hunt, our administrative manager. So thank you. Good luck, Joe.
I'm here, which is annoying.
So hello, welcome to the over 100 people who are in the Zoom. I guess this is the nature of things these days, is that we are bigger than ever before, but not all necessarily face to face. My name is Joe Koresh, and I do cardiovascular epidemiology. And it is a pleasure to see all these huge cohorts presented. And it's actually interesting looking at them, that it's both a wonderful thing to be here at Hopkins so that all these opportunities exist. And it's actually fascinating to see how things converge, right? So the biggest cohorts collect everything on everybody all the time, publishing all the time and trying to have a huge impact. So what do we do with ERIC? And ERIC is Atherosclerosis Risk in Communities. It was started by Moises Sklo. I help run it here with many, many people. One of the fun things about epidemiology is it's team science, but team science, when I heard it the first time, I was thinking, oh, that's kind of boring, but actually it's like professional sports. The best people on the field, beside the field, servicing the fields, really everybody at every position has to be at their best. So what does ERIC do? Right, so I'll outline a few things, an overview of the ERIC study data. I'll talk a little bit about that. We have visits, follow-up, stored specimens. Visit 10 is ongoing. It's a vascular study, right? And I've picked this because the blood vessels go everywhere. So it just means that when we started focusing on heart disease, it really goes from the heart then the kidneys, the brain, the muscles, diabetes, functional status, aging, cognitive status, dementia, and now we're even looking at hearing, et cetera. So it's become an aging study. So anything that happens to older adults, you have it. We keep renewing it. In fact, today I'm writing the renewal for the next 6 years. It's an open collaborative model, open, wide open. So we're over 2,700 papers. I think a lot of the other studies are as well, over 1,000 authors, many ancillaries across many institutes, and we share the data across many repositories. There have been various, the ERIC neurocognitive study is one of our major studies now. We've gotten interested in the idea that vessels don't only dictate disease in the heart, but also the brain, and that's a preventable form of dementia. If you control your hypertension and diabetes throughout your life, you are less likely to have dementia, and that is now in national recommendations, and that's one of the things we do is work towards recommendations. We're in many consortia, and I'll show you some things about the next generation. So these are some of our participants at their 30th anniversary visit here in Jackson, Mississippi. I'll do a whirlwind. We still have 3,000 hospitalizations per year, and these are some of our pioneering leaders. We still have mortality. Now it's shifting to ages over 85, but also some deaths 65 to 85, and then deaths before the age of 65. So really it's a life force perspective over 3 decades, which is probably true of the other cohorts as well. This is the type of thing to show you that we collect data on a wide range of things, on a wide range of visits. And you heard about the various domains. We cover a lot of the same domains as everybody else. The secrets to success is often an amazing staff that's dedicated. They know the participants over decades. Our field center is in Hagerstown, Maryland, and this is me demonstrating our newest retention gadget, which is a robe warmer. Post-COVID, we've decided we need a VIP experience, right? Otherwise, why would people leave their house? This is someone demonstrating one of our newest technologies. Liz Selvin has, I think, 3 R01 studies to look at continuous glucose monitoring. You put this little device and you get readings for, multiple days. We now have multiple wearables and trying to integrate data collection. I'm looking at Jennifer Schrack, who's looking at actigraphy. And then the newest renewal will have 2 different sleeping devices, one on your head, one on your wrist, and then we'll compare them. And then we're trying to get 90-year-olds to wear Fitbits. Omics, like everybody else, the world's enamored with omics. I did lots of omics stuff over my days. You hear the tone in my voice. I think some of the simple things like getting on a scale, weighing on yourself, tons of information. If you're not excited by that, we have millions of data points on genomics, epigenetics, metabolomics, clinical chemistry, proteomics, over 150 million measurements. I'll show you a couple of things on proteomics because I like data. So I did a webinar. You can look at this webinar on this. It's interesting. Some proteins, you measure them by targeted and untargeted, you get a great correlation. Some things you get 0 correlation. So it's an interesting world, this world of proteomics. You see things like PSA, which should be higher in men than women, but every once in a while you see women with measurable PSA levels. Is that biology? Is that trans? Is that mistakes? You can learn lots of things. This is Keenan Walker, who's done an amazing job going from a postdoc to writing JAMA papers to becoming probably arguably a world leader in the proteomics of dementia. And now he has his own research group at the NIA right down the street. And this is an example of the work where he found that 2 proteins likely play a causal role in dementia. And the types of ways in which we look at that is we look at it in the ERIC study, we do internal replication, we do external replication in other studies, we do Mendelian randomization to look at causes, and then we do pathway analysis, and we do candidate proteins in brain tissue to sort of do validation. A lot of the other people do that. Epidemiology has evolved a lot, and we do things like volcano plots to show which proteins are related to dementia, and elegantly he's looked at within 15 years, more than 15 years, and different pathways. He's also made suggestions for pathways and ways to look at prevention. So now he's implicated proteostasis, immunity, synaptic dysfunction, and extracellular matrix organization in dementia, which is a huge challenge. We have a whole cancer study here led by Elizabeth Platts and Corey Jishu. We've integrated hearing studies, and we've actually leveraged the study to a clinical trial whose results should be reported this year. We're closing the data now as to whether hearing aids will causally reduce the risk of cognitive decline with implications for dementia. Is it just comfort or is it really biology and necessary? So as a summary, Eric, we hope we're doing innovative science. We hope we address important topics. We look for timely execution. We're a fully distributed model, so every center has all the data. And the students, once you do more than 2 papers, get a password and access to network drive. You get access to everything. But there is an order to things. There's a publications committee. We review 20 publications every month. We approve over 200 papers a year and publish about 180 a year. So you're welcome to join us. Any topic that's related to aging and vascular disease, chances are we can do it. Thank you.
All right. Okay. Hi, everybody. So good to be with you. And for those in the room, if you guys have questions after this, just come up and ask us. Grab us while we're still here, because it's good to see you all. Thank you for being here. So I'm Carrie Altoff. I'm an associate professor in the department, but also did my MPH and PhD in the department. So I've been here a long time. And I co-lead the North American AIDS Cohort Collaboration on Research and Design with Richard Moore. It is a part, it's the North American region of this global consortium that follows almost 2 million people with HIV all over the world. We also have collaborators in Europe, but we don't directly, they run their own show and then we collaborate with them in different ways. But it's a whole wide consortium. We do have some students from Hopkins working in the consortium now. The NA Accord is concentrated in the US and Canada. We love maps, don't we? Oh my, do we love maps. So this is the NA Accord. We have participants from all over the place. We have both clinical and interval data. So the Max and the Ys, the ALIVE study, a couple of other studies of hemophiliacs from many years ago. They are all in the NA Accord. We also have a whole other group of studies which are clinical cohorts. So the data generating mechanism there is healthcare seeking behavior. And so we follow them predominantly through their electronic health records. So both clinical and interval cohorts, we follow about 180,000 people. We have over 1 million years of person years of follow-up. Median time of follow-up is about 4 years until they hit our loss to follow-up definitions. But it's important to note that these are predominantly dynamic. You heard some of these interval cohorts, they reopen, they recruit people. The clinical cohorts are always open. So they're asking people to join whenever someone comes in new to an HIV clinic. And so we follow them for a medium of 4 years, but we never kick anybody out, right? When they come back and we see them. So we complete a lot of tasks. This data is daunting, so don't be afraid. We complete a lot of these tasks where we take the data from these different cohorts who collect it with their own protocols. We have a data management core out at the University of Seattle or University of Washington in Seattle. They take the data, they harmonize it to our standard. We go back and forth a couple of times there. Then the data come here to the Epibiostat core in the department. We do a lot with metadata or cohort level data, as well as individual level data to create these analytic ready data sets. We feed this back to the cohorts through reports for that final phase validity check. And we run it all through co-investigator participation across different groups. We have 2 ways you work with us. Either we can help conduct the analysis through the Epibiostat core if it aligns with the parent aims of the grant, or at least half of our projects now pushing to 70–75%, we share data externally. There is an approvals process to nest a project, as with all of these. Actually, we all use the same process for approvals, pretty much, initially established by MaxWise, and we create a bunch of scientific products. So in this study, which was with ECHO collaboration and Katie Lesko led it, it's how we walk through and you actually nest a study in some of these studies, right? Because if you want to come to one of these studies, you nest your own study in here. And so there are selection criteria and different things you will put onto the study, and you will use the participants who have the data you want in the time you want it in order to conduct your analysis. So the gray stuff we do, stuff below the blue bar, this is what you would do with your research nested.
So we do have three predominant aims. As Joe said, everything is kind of an aging cohort now, and that's very true. So we focus first on the timing and outcomes of HIV treatment with specific reference to the HIV care continuum and ending the HIV epidemic in the US, which is a national priority. Aging with HIV, we have a lot of different outcomes, some validated, some are operationalized. We have a bunch of mental health diagnoses and conditions that we've just finished creating analytic ready data sets for, as well as multimorbidity. We also studied the co-infections, as Shruti alluded to, that are very common, hepatitis C and B. And then we have methods development. So how do we pull this individual level data, but use the cohort level data to make sure we're doing that correctly, and so on and so forth. Nested within this cohort, we have over 400 trans women. And in this lovely study, which was Katie Lesko's approach to looking at the HIV care continuum in a continuous time way, we were able to compare trans women to cis women and cis men. So 482 is a pretty large sample size of transgender women, and happy to have NA track as a nested subcohort. We also collaborate a lot with modelers. We provide a lot of data estimates and data functions. This is a model that I work on with Parastu in the department. And what we do is we use data input from both the CDC and the NA Accord to predict the age distribution of persons living with HIV forward in time. And then we overlay all of these different comorbidities and risk factors to really get at the essence of what it means oftentimes to age with a chronic condition like HIV. It is messy, it is medically complex, there's a lot going on. And then we reflect that by stratifying those different conditions into the 15 subpopulations that are predominant, because with HIV, there really isn't a one size fits all anymore. These are different subpopulations that create the population of persons living with HIV in the US. Within the NA Accord, because we have these strong collaborators, we were also able to quickly pull together the CIVITS collaboration during COVID, where we had 4 clinical cohorts and the MaxWise. We were on the phone biweekly. It was really fun, wasn't it, Amber? We were all a little loopy during COVID, right? It was frantic, but we were able to come together and think through a number of challenges to how do we move this data in a real way to make a meaningful impact. CIVITS resulted in 2 publications in JAMA Network Open that were produced this past year, and these, I just heard from a colleague, will be predominantly used for the guidelines for HIV that are going to be updated relatively soon, specific to how we protect persons with HIV against COVID. We're also putting our, I say this tentatively, our hat in the ring with post-COVID conditions. PCC, I think, is the new acronym. It was post-acute sequelae. It's long COVID. That's what everybody knows it as, right? But working with the team across the street, so Ann Parker and Alba Zola are the clinicians who have been running the long COVID clinic across the street. And so Kelly Gibo and I helped them establish their clinical cohort and get the data moving for them through with the amazing help of the EPIC team across the street. EPIC is the electronic health record. And then we're also looking at a bigger swath. So we're looking at individuals who we believe were seeking care at some time before COVID at Hopkins and then had a COVID test prior to the kind of flooding of the market of at-home tests. And we're going to look at these individuals to better identify and continue to work at this very complicated compilation of syndromes known as long COVID. So those 2 data systems are in the works, and data will be available in about 5 to 6 months. So I have a lot of cohorts, right? There are a lot of things based. I agree with Shruti, having something Baltimore based is very important. Then we also have all of these global cohorts. So if you have interest in nesting anything across here, let me know. We've had a lot of success with our students. We have a study staff, a group of professionals that also assist. But I think one of the things, as you're thinking through all of these, this must be like a fire hose for a lot of you thinking about thesis work. But just remember that, you know, I have worked in the ALIVE study. I did my thesis in the WISE, and I've worked with the MAX data. I mean, we know these different studies, and if you have a study question that we know would work better in a different study, we will direct you to that, right? So I think that's important to know. So if you haven't fallen in love with a population or a disease or an exposure, you can go to one of these studies and say, how can I help? Or you can be driven by your love of population disease exposure and go to one of these cohorts. And if you're not quite, your question doesn't quite fit there, we will direct you to the right group. So that's all I have. Thanks.
Okay. Hi, everyone. I'm Jennifer Schrack. I'm an associate professor in the department, and I'm here to talk about the National Health and Aging Trend Study. I'm happy to see aging as being so well represented across so many of these studies today. So what is NHATS? NHATS is a multidisciplinary consortium. It's a longitudinal panel study of disability trends and dynamics in the United States, and it was started in 2011. Judy Casper was the PI. She was a professor in health policy and management. I took over about a year and a half ago. It's run as a cooperative agreement with the NIA, so it means we're a UO1. And it's led primarily here at Johns Hopkins and also at the University of Michigan. I have a co-PI, Vicki Friedman. And then we are in the field. We don't have any clinical sites. We deploy somewhere between 150 and 200 data collectors through Westat, so a company in Rockville that manages our data collection for us. We get input from a lot of interdisciplinary investigators. So what I mean by that, we have 2 advisory boards. We have a steering committee and we have a data monitoring committee with experts in public health, demography, sociology, public policy, economics, geriatrics and internal medicine, and physical medicine and rehabilitation. So we really are not just epi, we're not just demography, although that's who runs it, but we get a lot of input from a lot of people. The study design, we draw our sample, it's Medicare beneficiaries that are selected based on age and geographic location. We oversample Black, Hispanic, and individuals over the age of 90 because they have historically all been underrepresented, especially in aging research. We conduct annual in-person interviews, and these are done in the home by the data collectors, or I should say place of residence. So if people live in a facility, in a nursing home or a residential care facility, we do our best to actually get to them as well. Our data are released annually, and they are publicly available. Most of it, I should say, on our website, nhats.org. So you don't have to have our permission to use this. You do have to register, and that is mainly a tracking mechanism so we can report to NIH how many people are using our data. We also have restricted files. If you're interested in things like geography, geographic indicators, if you're interested in genetics, we have some genetic data. Those are on a data enclave that's run out of the University of Michigan. So MICTA, Michigan Center for the Demography of Aging enclave. You can get that. You need some IRB approval. You need approval from us, but you can get that. And then new, relatively new is our linkage to CMS data. So Medicare claims data. This has always been around, but it used to be that you had to apply, you had to pay for the data from CMS. It was quite expensive. What is new in this year is now available is this is free of charge. So there is paperwork involved. You have to have IRB approval. You have to have a data use agreement with Johns Hopkins and a data use agreement with the NIA. But once you have those things in place, the access to these Medicare claims data is free of charge. So you go on there, it's a secure data enclave. It's run through a company called Medric. You can go on there and link our data with people's Medicare claims data to look at outcomes. So our sample, so it started in 2011, like I said, just over 8,000 individuals. And so that is the blue cohort. So the blue bars are the original 2011 cohort. It was replenished in 2015. That's the green bars. So after it got down to about 4,000 living individuals, we replenished, got back up to over 8,000. It's now gotten down. We were supposed to replenish again in 2020, but because of COVID, that was put off. So we replenished some this year in 2022. We're now back up to just over 6,000. We're going to replenish some more in 2023 and try to get to 10,000 older adults. We are growing. We are continually expanding, especially as interest in Alzheimer's and dementia continues to expand. So what kind of data do we have from these people? So we have what's called the sample person interview. So this is our annual interview that's conducted, like I said, in their place of residence. This is conducted with a participant, or if they're unable to do it on their own, about 6 to 7% have a proxy respondent. This interview takes about 2.5 to 3 hours. It depends on the person's capabilities and their condition and their ability to respond. The interview is pretty extensive, a lot of questions about ability to take care of themselves, ability to do things,
mobility, capacity. And then interestingly, we don't stop there. A lot of studies end at disability. We also get and talk about accommodations. So you have a disability, do you have accommodations that allow you to continue to live independently? Do you have a caregiver? And we try to get at those accommodations that people have as well. We do some objective measurements in physical performance, the short physical performance battery, grip strength, and then we also do a peak airflow test. We do cognitive assessments and objective and also iPad based, and we have an algorithm for possible or probable dementia. And then in 2021, we added hearing, vision, and accelerometry in a subset. So hearing and vision in everyone, it's an iPad based hearing and vision assessment. And then accelerometry in a subset, just under 800 people. We're going to expand that, or we plan to expand that in the next grant cycle. If people live in a facility, we have a questionnaire that is conducted with a staff person at the facility that tries to get information on the level of care offered to the participant, their services, and also the costs associated with those cares. And then once people pass away, we do our best to conduct a last month of life interview with a proxy respondent or someone's representative to try to understand what their last month of life was like. So to learn more, there's a ton of information on our website. So this is the NHATS website, just a screenshot of it, where you can see all our user guides, our technical papers, the instruments. You can go on and see everything that we use to collect this data, the data collection materials. And like I said, if you register, you can download over 10 years of data. If you want the restricted files, you have to apply for those. There's also a link to our sister study, which is NSOC. So National Study of Caregiving. So if we find out people have a caregiver, an informal caregiver, unpaid caregiver, we do our best to interview those people and try to understand the extent of the caregiving and what people need. And that's pretty much it. So thank you.
Yes. There we go. Great. Well, thank you, everyone. And as it goes with these cohorts today, I think I'm presenting the baby cohort of all of these studies. So this is a study called the American Cohort to Study HIV Acquisition Among Transgender Women. We also go by the LIGHT cohort, which was the name of the RFA that initially funded this study. And our community advisory board members loved it and changed it to Leading Innovation in Transgender Health and Empowerment. So to give a little bit of background, this study came out of the knowledge that there is a very high burden of HIV among transgender women in the US, particularly among transgender women of color. And despite that, and despite historical contribution to research and trials, there really hadn't been a prospective cohort of specifically of transgender women in the US to date that could really explicitly inform prevention strategies and monitor the HIV epidemic among this particular group. As a number of colleagues and I were discussing this, thinking about a potential cohort for transgender women, there was an RFA released by the National Institute of Health called the LIGHT RFA, and that was really focused the acronym for that is Limited Interaction Targeted Epidemiology. And they were really interested in moving from traditional facility-based cohorts towards more digital cohorts. So really explicitly, completely remote cohorts that could collect biologic specimens, conduct survey questionnaires using technology. And this was really built on the assumption that there's a high level of ownership of smartphones, cell phones, tablets, access to internet across the US. And that's true, we recognize that that's true, but technology access can be inconsistent. And so we applied to that particular RFA and we pushed back, we were a bit stubborn, pushed back and said, this will be one of the first cohorts of this population for whom there's really high levels of research mistrust. And so we actually proposed 2 modes, and I'll tell you a little bit more about those, but really tried to recognize at the beginning of this study that there are still technology vulnerabilities in the US, and that exclusively digital methods have the risk of potentially excluding people who are truly vulnerable to HIV acquisition. So ultimately, we aim to develop this multi-site cohort, and this was in 2017 when we submitted this proposal. It was funded and we launched in 2018. It's community engaged research. So the study has the involvement of students, staff, and faculty who are trans-identified across all of the institutions and sites in which we work. And we also have a community advisory board as well who contribute a lot of input throughout the study. We developed a mixed mode cohort. So there were largely two modes within this. One that was a technology enhanced site-based mode. So spanned 6 cities in Eastern and Southern US. And by technology enhanced, we required participants to come in person to our sites at baseline and every 12 months so that they could build relationships with our staff, build trust, et cetera, become familiar with the technology. We developed this hybrid app. So it's a native app that can be installed on smartphones, but also available through a web app for people who don't have smartphones as well. And so after those baseline and 12-month visits, participants who even in the site-based mode could still participate remotely if they wanted to through our app and through the use of oral fluid HIV testing. We had a second digital mode that spanned 72 cities in Eastern and Southern US, and they were matched in population size to the initial 6 cities. Participants complete regular survey questionnaires. Those in the site-based mode also complete SCI testing every 12 months. They also provided stored specimens. And we provide referrals for confirmatory testing and other services. So as I mentioned, we launched in 2018 and the vast majority of participants were enrolled by 2020. We still had some people who enrolled up into August of 2020. And that was actually quite useful for us because people were at that time, participants were accustomed to our technology and these remote specimen collection methods. So we're able to actually quickly transition to really completely digital methods for a while during the early phases of the pandemic. Overall, we enrolled 1,312 participants in the cohort, 587 within the digital mode, and then 725 in the site-based mode. And what's been really nice about that as well is that within our particular study population, participants are actually quite mobile. And so when someone left a city, moved out of a study city, they were still eligible if they stayed in Eastern and Southern US. They could participate remotely through the digital arm. So we had several participants who transitioned between site-based and digital modes or from across multiple cities within the digital mode arm. As was our main objective to really understand more about patterns of HIV risk and HIV incidence, it's provided a lot of opportunity to really understand the unique behavioral risks for HIV among participants, and also really monitor uptake of pre-exposure prophylaxis, which is highly effective in reducing HIV acquisition risk. So this was a paper that was published by a doctoral student, Erin Cooney, that focused on understanding more about those longitudinal trajectories of both behavioral risk and PrEP use and adherence over time. And again, as with our outcomes of interest, we were really focused on understanding HIV incidence. So we recently completed that analysis and found that across over 2,730 person years of follow-up, there was an HIV incidence of 5.5 per 1,000 person years. And probably not unsurprisingly, based on what we know about the epidemic in the US, this was much higher among Black participants and those living in the South. And actually within the 2 cohort modes, also much higher among participants in the site-based modes versus those in the digital cohort arm. Over the course of follow-up, we did also observe several participants who had passed away. And so we felt like, despite our objective to focus on HIV, that we couldn't really think about and talk about HIV incidence and prevention without also considering the factors that put people at risk for premature death. And so we looked at mortality as well and found a rate of 3.3 per 1,000 person years, and particularly higher among Latina participants. And really those forms of premature mortality were largely attributed to homicide, suicide, overdose. There was really only one cause of death that was a natural cause of death among those participants. And I think importantly in all of this, we found that there were a lot of overlapping indicators or predictors of HIV acquisition and premature death. And so it's an opportunity to think about how we can use prevention strategies to also improve the health and well-being and survival for trans participants. When we started this study, we recognized that, as I mentioned, there's high research mistrust, but for a lot of people in the community, while people recognize and think and consider HIV as an important factor to study, it's often not people's priorities. There are other issues around survival, violence, and other main factors that are really the priority that the community wants attention to. And so we've spent a lot of time working on developing measures. A lot of this is more in the social epi realm, but this is one example. This is a paper published by Sarah Peitzmeier, who spent a lot of time developing and validating a tool to measure trans-specific forms of intimate partner violence among our participants. And we also looked at longitudinal risk of violence victimization and incident violence victimization, which you can see is quite high, and particularly among those 18 to 24 in our cohort. Similarly, incarceration is a main concern given the hyper-incarceration of transgender women in a lot of areas. And so this is another study that was published by Tonya Petit looking at incident incarceration over time. Our study under the Light RFA has actually just is ending this year, but we've had another opportunity through the NIH to continue the work with the cohort and actually expand it nationwide. So the Light study will continue under the name ENCORE, Enhanced Cohort Methods for HIV Research and Epidemiology. We're continuing with this hybrid model, recognizing the higher risk that we observed among site-based participants. And so this will expand, again, the cohort nationwide. We'll continue to have hubs that can provide site-based support for participants who may need additional support in completing questionnaires or providing specimens. We're building more research in around syndemics and analysis of how syndemic experiences affect HIV incidence. We'll be bringing in contextual data. This will be led by Sabria Linton and the Department of Mental Health, looking at how other population-based characteristics affect one's individual HIV risk. And then also mathematical modeling with Anique Borquez and UCSD to understand what are then optimal prevention strategies. So there's a lot of data, a lot of opportunities to be involved, and just to recognize the large group of people that are making the study happen, and certainly the participants as well. Thank you.
Right, I know some folks need to get to class, but others are invited to come down and ask questions for our speakers that can stay. And thanks to everyone who joined us on Zoom. Students can email me because I think there's no way I can do the microphone. They can email me if they have questions, but I don't see any. Yeah, thanks everyone.
Hello, there's no questions in the chat, basically just thank yous. So
Okay,
thank you,
great.
everyone. And we'll close the Zoom online, and